Paula Tran, and I worked with Lianna, a Ph.D. candidate, and Dr. Daniel Barta, and we are here to present to you the variable phylogenetic distribution of Wormian bones within mammals. So I know what you're thinking, Paula, what exactly are Wormian bones? Wormian bones, also known as intrasutral bones, are irregularly shaped bones that develop from different ossification centers. Cranial bones from the anterior skull are derived from neural crest cells, while the posterior skull is derived from the mesoderm. However, the interparietal bone, which is posterior to the parietal and superior to the occipital, is derived from both the neural crest cells and the mesoderm, differing from its neighboring bones. As you can see in figure 4 with my laser pointer, the blue color-coded section is from neural crest cells, while the pink is from the mesoderm. The interparietal here is color-coded with both, indicating, again, it's both from the neural crest cells and the mesoderm. So here, back in figure 1, the yellow bones represent Wormian bones that we've seen in humans. So there's really no concrete explanation as to how and why intrasutral bones form, although there have been some genetic and environmental factors that have been proposed. First, with brain development. Brain development can affect the signaling of skull formation and the suture closure timing. Parts of the embryonic brain can be associated with different parts of the skull formation. Second, mechanical stress, which this was in horses, have shown that the formation of Wormian bones kind of reduce that strain when there is stress. Another hypothesis is that the restriction of bone growth can lead to intrasutral bone formation. For example, when one suture is stuck, Wormian bones may form in other sutures to reduce that strain. Intrasutral bones have been highly correlated with central nervous abnormalities in humans. So the incidence and location of intrasutral bones vary among different species. We conducted a detailed literature review to find the incidence of Wormian bones and what species they usually happen in. So Wormian bones can have a lot of synonyms. You'll find that it could be intrasutral, like I've said before, or fontanelle. Even some Wormian bones have the location in its name. And so you'll find examples of the bregmatic bone or the Tyrion bone. So the categories of the Wormian bones includes the bregma, coronal, frontal maxilla. There's 4 different interparietals, just because the interparietal starts from 4 different elements and slowly merges into 1, 2, or 3 during embryo. Then you'll find the lambdoid, the metopic, the nasal, Wormian bones found in the orbital region, Tyrion, and sagittal, all of which you can see on figure 2 right here, showing the different categories of sutural bones highlighted, with the bregma, for example, being the black star in the middle, and then the metopic being the light pink suture right here between the 2 frontals. Moving on to the results, the bregmatic bone, the black right here, has shown a consistent occurrence throughout mammals. When the bregma bone has been identified, there hasn't been many incidents of the frontal maxilla, the light blue, or any Wormian bones in the orbital regions found within the same species. Another result found was that instance of a Wormian bone towards the front of the skull, so metopic, nasal, and corona, have rarely been identified together in the same species. Supernumerary bones in the Tyrion right here, so the light orangey brown, has mostly been recorded in primates with an incidence found in the Canis species. The bregmatic and the interparietal bones, right here with the 4 different elements and the black boxes again, among species, are found more often compared to smaller anterior and intrasutral bones. These results are consistent with the hypothesis that Wormian bones anterior to the coronal suture may develop more variably compared to Wormian bones found posterior to the coronal suture. And this could be due to there is more variability in neural crest cell development compared to mesoderm-derived bones. Alternatively, the variation in intrasutural bones among mammals can result from different interspecific variation of the shape and size of the underlying brain region itself. Some variation may be an artifact or coincidental finding of an intrasutural bone recorded by previous authors, and this will be addressed in the future more exhaustively when we survey individual species to assess how often each Wormian bone is assessed within the species. We would like to thank OSU, the Federal Work Study, Dr. Smith and Dr. O'Brien for their help. And last but not least, I would like to wish everyone a great day, and thank you so much for listening to me talk about Wormian bones.